Hello, everyone, and welcome to today's webinar, Automated Microbiology Plate Reading with Artificial Intelligence, introducing the Clever Culture Systems APAS Independent Instrument. We are pleased to announce that the APAS Independent System has received the CE mark under IVDR regulation, additionally to the MRSA module this week. I'm Christy Jewell of Labroots, and I will be your moderator for today's event. We encourage you to participate today by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. We have two speakers for you today, Dr. Ulrike Schumacher and Dr. Stephen Giglio. For complete biographies on our speakers, please visit the presenter window at the top of your screen. Dr. Giglio will be speaking first. Welcome, Steve. You may begin your presentation.
Hello and welcome to the seminar today. Look, my name's Steve Giglio. I'm from Clever Culture Systems, and we're going to be talking about artificial intelligence and how it can help the micro lab. This presentation will take you behind the scenes around AI and how we do what we do at CCS, and hopefully you find it entertaining. Next slide will be a disclaimer about myself, and we'll jump straight in. Muffins, right? So why am I talking about muffins? Well, is it muffins? If you look at that picture very, very closely, I think you will see that there are indeed some muffins and maybe something else. So if we just zoom in, we see muffins and chihuahuas. So this is a really interesting problem that a lot of people in the AI world have been trying to solve. And there's a number of systems that exist that use the images and the various types of AI technologies to try and differentiate a muffin and a chihuahua. So I think if you squint your eyes and have a look at it, you'll start to see that they start to look similar. Is it a blueberry muffin? What type of muffin is it? What type of chihuahua is it? And there's a lot of published data around this specific challenge and really the ability of AI systems to differentiate it. So I'll just leave that thought with you for now, and it will come into context a little bit later on. So what is AI? AI is a field of computer science and psychology which is intended to replicate human intelligence in a machine. Now, it sounds fairly straightforward. Everyone is aware what AI is. Everyone's used ChatGPT. Everyone's gone on Google AI. The field actually really started in earnest around 1950. There was a person called Alan Turing who is the father of AI, and started to really think about AI and some of the philosophical questions around what it can and what it should do as well, very, very importantly. He started to document his thoughts, and not long after that, there were machines, computers, very, very basic computers that mimicked playing chess and playing checkers. And it turns out that humans were still very good at playing chess and checkers, and the computer systems at that time really couldn't keep up with the human brain. And there's always been a strong link with AI and biology. And when you think about how the brain works and the neural networks that are there, there's neurons, there's billions of neurons, there's decisions that are made by the human brain, which is very, very quick and instantaneous. That still hasn't been replicated by any computer at the moment. So when you think about how quickly humans make decisions and how quickly computers make decisions, humans still make decisions quicker than computers. No one really knows how the neurons work. I mean, I think there's a lot of information around the physiology and the synaptic charges that are there to transmit information and make all the neurons fire together. But AI is really trying to mimic that. If you look at the diagram on the bottom right-hand corner, AI is looking to process large amounts of information in a way that's repeatable and predictable. And so there's a number of features or attributes that we look at for any specific problem. And based on probability and likelihood, there are decisions that are made based on decision trees. And this is not dissimilar to how any person works in terms of framing a problem and trying to solve a solution. There's different ways to do that. And AI is just really trying to replicate that thought process and be assistive and not substitutive. So, AI is not looking to replace anyone. It's here to help with very specific things that humans do struggle with. One of the big drivers in AI is data quality. We're starting to really understand this with some real earnest around it as well. And the main factor driving any quality of a AI system or a machine learning system is the data. And so the old garbage in, garbage out is relevant here as well. If we look at the diagrams on the right-hand side and going back to the muffins and the chihuahua problem, you can see that what needs to happen with any AI system is you need to segment the images and look at the inputs. And so it's very clear that most of those inputs are right. And if you look through what's a muffin and what's a chihuahua, you'll see that the one highlighted is actually a chihuahua, but it's labeled as a cupcake. Now, if this data is used as part of the machine learning system, then there's quite a bit of influence on the incorrect data. And so there'll be a level of confusion between what is a muffin and what is a chihuahua. Importantly, when you develop a system using AI, it's not normally transferable. So if you're looking at developing a system of AI on one particular hardware which takes images, it's not going to be transferable to the next. So think of a microscope in your laboratory. There are many brands of microscopes. If you were to put a camera through the top of the microscope to try and take an image, then there's different influences of lighting, influences of color illumination, different lenses and quality of lenses, and they all have a very significant impact on the acquisition of the image and therefore the way the data is interpreted. And so it's important to have very broad training scenarios to capture all those variations. We want to have data that is representative of the real world. It needs to be controlled and it needs to be authentic. And so there's a lot of work and a lot of people who are now data cleansers. There's a lot of people whose sole job in AI companies is to clean the data and to know if the data is real or if it's not. And sometimes it's hard if you're not involved in that very specific segment of work that you're doing to know if the data is real or not. And so from a microbiology point of view, you want microbiologists to drive the fact that data is real or not. And so, going back to garbage in, garbage out, the data is absolutely critical for any successful AI system. So if we look at those two images on the left-hand side, and you need to remember that this is what computers do. They look at pixels and segmented images to make decisions. I think if we looked at that objectively, it might be a coliform, there might be something else. It could be a small Pseudomonas. When you look at the group of organisms that it's come from, one is in fact a coliform and one is in fact a Streptococcus. So these are images that we use to develop the AI, and you can see there's a cluster of different types of coliforms on the left-hand side, and on the right-hand side there's a different cluster of Streptococci, and there's elements of overlap there. And this is really a really important fundamental principle of any AI is confusion. So this overlap that occurs in an AI system is important to recognize that it exists, but also it's a limitation of the technology. And I think we've all had bench experience around showing people a colony on a plate or a growth on a plate, and not everyone would understand or get the correct answer. So is it an E. coli? Is it a Citrobacter? Is it a Enterobacter? There's different ways that humans interpret that, and this is part of our challenges as an AI company to develop that common understanding through microbiology and AI. So AI is here to help. It's not here to take our jobs or do anything like that. And certainly what we've developed here at CCS is the APAS independence, which is definitely an assistive technology. It reads and interprets plates. It's the only auto-verified imaging system that is cleared by the FDA. So there are claims that we make with our technology that does not need any user intervention at all. It's obviously very easy to integrate. It's a modular. It's on wheels. It rolls in. High level of performance in terms of clinical data that supports its use, and it's an affordable stepwise automation that can be introduced into laboratories. So what do we need to consider when we're looking at any AI system to implement into the microlab? And we kind of need to ask ourselves, well, why hasn't this really penetrated in earnest? When you look at ChatGPT, and I think they had maybe several hundreds of millions of users in several months. Why hasn't that happened in the lab? There's essentially no scalable automation in labs that you can tag onto. You can't just use your existing infrastructure and put a camera in an incubator or on a microscope to now have an AI capability. The amount of capital available in microlabs is traditionally very low. COVID certainly changed that perception amongst administrators, and I think the administrators now realise that the microlab and general pathology labs is an important part of the hospital system. People don't know when to jump in. There's still trepidation around what AI is and when is the right time to do that. And sometimes the expectations are misplaced. Microbiologists need to understand what the system is and understand what the limitations are. And so there's obviously a body of evidence that people look to take that on, and it just takes time. So providing digitisation and standardisation is the key for culture plate reading, but the lack of data sources, like we discussed, limits the advance. So we at CCS control all the data, and so we very much know what we like. The problem that we're trying to solve is generally unclear. Microbiology is very much an art, and I think if you look at the way a urine is processed in any one lab across the globe, every lab has a different way of doing it, and it's certainly been my experience having looked at about 40 labs across the globe in terms of how to process a urine. It is significantly different lab to lab. You wouldn't have thought so, but it is. There's also a priority within the lab. What's more important? Do I want a culture plate reader? Do I want TLA? Do I want front end of TLA just to do streaking? Do I need automated sensitivity testing? So where do we prioritize the use of AI in the lab, and understanding that the culture plate reading is a highly skilled task, and that is an important job that still remains. And what AI is serving to do is serving to augment that. So removing a lot of the negative or no growth plates from urines or from infection control screens. That is the job of AI in the microlab. So just a bit of behind the scenes of what we do here at CCS. We have a group of microbiologists that work with engineers and software people, and essentially we're trying to turn our thought processes into code. So when I say our thought process, us as microbiologists, and that's certainly my experience as a microbiologist with 30 years experience and managing labs, it's all about, well, how do I think and how does everyone else think, and how can we be assistive with that? And so if you look at the image on the top left-hand side, we've just got a urine chromogenic plate. Moving to the top right, this is essentially like Photoshop. We paint the dots. We call these labels. So these are unique labels to different types of colonies. So this is now us training the algorithm. And different colors mean different things because they're different colonies. So there's a fair bit of logic that goes around how we do that, and the decisions for what a colony is or what is confluence or semi-confluence. And this is all driven by the microbiologists. So we work quite closely with software people to do this. The end result, if you look at the bottom two images, is this is a VRE plate. You can see that the image on the left is has got Sharpie marking, it's got labels, it's got imperfections on the agar plate. The right-hand image on the bottom then is the APAS assisted, or is the APAS image, and it just shows you, well, it eliminates the labels. It's picking up colonies on the edge of the plate, and that is what is driving the result. So one part is to get that per pixel classification from APAS. The next job is, well, what do we do with that information? And that's the role of the microbiologist to drive a lot of that as well. Excuse me. So I came up with a term called a microbiologist in terms of algorithm development, and again, this it is a very, very difficult thing to do. I think as microbiologists, we tend to ignore the level of complexities that we just routinely do in our mind, and when you speak to people outside of the field, it's not obvious what we are actually talking about, and so it's quite the job to linearize the thought process to drive the input into a code. So when you think of what a computer code is, it's essentially zeros and ones, and so the information is very literal and very binary. And when we go through the process of developing algorithms, we really need to dissect that thought process critically and objectively. The good thing with working with multiple people, like I said, the engineers, the regulatory people, the software people, is that there is a reflective element where we start to think about, well, what did I actually mean by saying, this is a coliform or this has hemolysis, for example. It sounds very, very simple to just say it as a microbiologist, and as a microbiologist, it's understood. Translating that to code is very difficult. So we go through quite a vigorous process of developing the algorithm. On the bottom right, you'll see a traditional V model of product development, and you start with defining what the project is. You get to the bottom of the V, you make your algorithm, and then heading back up the top of the V, you start to test and integrate it. Now, there's a lot of work that goes into that. There's a lot of challenging assumptions. You're revisiting the use of the algorithm, you're redeveloping it, you're doing a lot of testing around it. So there's a very iterative process around the development of the V model there to prove that the system works. You can't actually predict how the system works unless you test it. So you have an idea of how it would work, but unless you do some very specific testing, you can't prove that the technology works. And it's a really important point. It's not something you could just go onto Google AI and really try to test. There are very specific ways to do this, and also a lot of regulation around that. So, kind of touched on this a little bit earlier. There's the data is the most critical bit. Using a single source of data will produce a limited model. So if I wanted to look at the bugs that grow in your lab, I could produce a model for the bugs that grow in your lab. It doesn't necessarily mean that it's going to work in the lab in the next country or even next door. It depends on the demographics that go in. So we use a very broad range of data sets across the globe. We do a lot of different things to try and make the AI sensitive to the broadness of changes that occur in labs. And we go through a very kind of hefty testing process to prove that works. And that's the proof is in our regulatory clearances for that. One of the biggest things to deal with an AI system is interference. There's a lot of things that are on plates, and you can see some images there of the plate artifacts. So there's injection molds, there's dot matrix printings, there's in the inoculation effect where you get pigmentation from cross reaction of creams on plates. All of these things need to be trained out of the AI system because they do look like colonies, whether they be black colonies or little white colonies. We need to train that out of the system. The compromise is that sometimes you do get white colonies that are small, and that's now a challenge. Some of the other systems that use a time zero image, can overcome that challenge, but they don't overcome the challenge of the inoculation effect, which develops over time. So after all of that, just looking at looking at a McConkey plate and looking at the top lit image on the left, the middle the middle image is the bottom lit image. So from it's actually lighting from below the plate, and this is useful for hemolysis, but it also adds another level of definition and features that the AI systems can use. And on the right hand side, we have the APAS per pixel image, and you can see there's obviously lactose fermenting organisms that are pink that have been detected, and they're showing up as yellow. And we've got that one colony of a non-lactose fermenter that is coming through, which is circled, which has been accurately identified as well. And what I've added to the image is just a shadow of the label, and that's been removed. And if you look at the 11 o'clock position or the 10 o'clock position on the McConkey plate as well, there's also the plate location mark that's also been removed. So it's really cool stuff to be able to do that and develop it. So I feel fortunate to be able to do that. Ultimately, the proof is in the results that we get and our regulatory clearances. And what I will present here is MRSA performance. We have IVDR pending for our urine application, and that's some data we may present if it's available. The performance of MRSA across multiple sites across the globe is pretty good. When you look at the sensitivity, we have a sensitivity of 100%, and with APAS, one of our big strengths is really the way we can get very high specificities across these testing plates as well. So, our lowest specificity there is 94%. Our highest is close to 98%. So, really high sensitivity, really high specificity. And we know that APAS has proven microbiologists wrong on several occasions. So a lot of these studies demonstrate that humans do miss the needle in the haystack, the colony of MRSA, that when you're screening, tens or hundreds of plates, you invariably miss them. So there's an example of that on the right-hand side, and this is a MRSA growing on a BD plate, and this was published out of Johns Hopkins in 2021. And you can see on the image of the full plate, it looks almost negative, but there is, if you zoom in, you'll see a morph colony there, which is a suggestive of MRSA. So when we look at kind of general workflow and impact on workflow, and again, this is an example of a generic urine system from a US study. We are looking to linearize a complex process. We can incubate the plates in the carriers. We can then run them through the APAS independence, and those plates with no growth or no significant growth can be sorted out. So when you look at the impact on workflow, there's no change in processing, there's no change in incubation time, but where it does impact is in that reading time. And it can be, depending on what your workflow is, there can be quite the drop in hours taken to read the plates. If you're in a lab that is 24/7, you can now start to have these systems kind of performed live and online rather than batching them. So, which is traditionally how work is done. There's you batch it for the plate reading shifts. There's an opportunity now where you can think, well, if you implement the APAS independence, you can release negatives as they become negative, with basically technicians to load and run the instrument. I'll just leave you with another thought. It's important to understand what the limitations are for any AI system. So, as I said, AI is assistive. We're here to augment the skill of the microbiologist, not here to replace it. There's a lot of years that you need to train for to become a skilled microbiologist. It's important to understand the limitations, and the confusion is one of the big things. So, looking at a urine chromogenic plate, what is enterococci and what is the Kisic group? I think when you get the context of all of those things together, you start to get a feel of what organism group is what. But if you look at those boxes that are highlighted, there are similarities across different organisms, across different groups. And so there is this natural tendency of chromogenic media to have this cross reaction or drift of colors, sizes, and shapes. And AI cannot really differentiate that 100%. It's a bit like the muffin and the chihuahua. AI will do a very, very good job for a large percentage of it, and that's AI's job, but it's not going to be 100% accurate 100% of the time. And I think it's important to really understand that, because from an implementation point of view, how do we work around some possible misclassifications? And that's a really important thing to discuss when considering this. So in conclusions, AI applications are rapidly emerging and are generally assistive. Really important. We're not here to take anyone's job. We're here to make life easier for everyone. There are strict regulatory positions on AI and applications, most of which still rely on human intervention for any growth. And that's really important when you look at the jurisdictions, whether it be the US or IVDR, there's different levels of criticality around what evidence is needed to prove, does a human need to intervene or not. Certainly our position at CCS is to make sure that we have as less human intervention as possible, which means there's a higher burden of proof. And that's the bar we've set. A lot of people think that algorithms learn as they go, or why can't they do that? Why can't we just have an open system that just learns on its own? It's not that easy. It is a fixed system. The regulators demand fixed systems. Happy for you to change what the system is, but they need to be purposely changed and retested thoroughly. So there's a fair bit of work that goes into that. We've talked about AI being suited to repetitive tasks, and infection control and MRSA screening is definitely one of those. And importantly, the removal of plates with no growth and no significant growth adds value to labs. There's a lot of things that goes on in the micro labs. We're all looking to free up some time. It's hard to get good people. It's hard to keep good people. And certainly being involved in something that involves AI is exciting for a lot of people we talk to, and that's the value of what staff determined to be something that they would consider to stay and do some extra work with. So in terms of staff retention, it's really, really important. Thanks everyone for your time. Feel free to get in contact. I'd love to hear from you. Thanks very much. Just as a late coming slide as well, we have literally got IVDR clearance for our urine module, so we can present this data now as well. Just summarising the urine analysis modules by region and by laboratory and by sensitivity. Just a real quick and broad snapshot of the diversity of the labs where we've tested our urine AI modules. And we've covered regions in the US, Australia, France, and obviously Germany. We've gone through several different types of agars. You can see the sample size there as well is also quite large, and the sensitivities are also very, very high. So as I said earlier, the testing of urine is not standardized. So it is a difficult task. The appetite for screening and auto-verifying is different in different labs. Some labs may want to release no growth, some labs may want to release no significant growth. And we know, especially with the chromogenic agars, that there are clear indications from Thermo Fisher and bioMérieux around the presumptive identification of E. coli, which turns pink or burgundy on those agars. And we know with APAS we have a very high predictive value on that identification. So look, I think I will just finalise my talk and just leave you with that thought. And I'm looking forward to hearing Ulrike talk about MRSA.
Thank you, Dr. Giglio. And as you mentioned, it is now time for Dr. Ulrike Schumacher's presentation. Dr. Schumacher, you may begin.
Okay, hi, fine. In this talk, I would like to share with you our experience with the EPIS independent system for MSA screening countries. Today, the microbiology labs have to master the balancing act between the lack of trained personnel on one side and the rising requirements of modern diagnostics of infectious diseases on the other side. So in this situation, we appreciate very much the possibilities of new automatic systems and artificial intelligence applications, provided they fulfill the requirements of sensitivity and specificity compared to the conventional microbiology methods, and simultaneously saving of working time. That are my disclosures I have to show. And then let me shortly introduce our laboratory and then outline shortly the conventional workflow of MRSA culture-based screening. The MVZ Labor Ravensburg is located in southern Germany near the Lake Constance, and we are providing laboratory and microbiology service for more than 100 hospitals with 12,000 beds, and also for more than 2,000 medical practices. We are processing about 3,000 samples in the microbiology daily, and although screening of MRSA and VRE declined slightly over the past years, today we are receiving more than 500 screening samples, and roughly half of them are for MRSA screening. The EPIS independence was invented in our laboratory in December 2020 after time for testing. The conventional MSA workflow, MSA screening workflow starts with inoculation of a patient's sample on a chromogenic medium, which is then incubated for about 20 to 24 hours. After incubation, growth of specific colored colonies are checked by a visual inspection of the plates, and suspicious colonies are further tested, and finally all plates are categorized as positive or negative. Because the vast majority of the samples are negative for MRSA, a lot of working time of technicians is wasted for looking on empty or clearly negative plates. So the APIS replaces the visual inspection by an automated imaging. By artificial intelligence, these images are then assessed as presumptive positive or negatives, and negative results can directly be sent to the laboratory information system for report. The EPIS instrument itself is a compact, not larger than a safety cabinet, and through its wheels, it's easy to change the location of the instrument. During the renovation in our laboratory the last 2 years, the EPIS changed its position in our lab several times without any problems. The EPIS is composed of 3 modules: the so-called input module on the left-hand side, in the middle the imaging module, and the output module on the right side. For a reading session, you can place up to 4 carriers into the input module, and each carrier includes 4 columns for up to 50 petri dishes. In the touch screen in the middle, you will see the displayed loading of the instrument. When the reading session is started, each single plate of a stack is then transported to the imaging module, will be scanned and immediately be assessed by the software. Corresponding to the assessment, the plates are then transported to the proper position in the output module. Presumptive positive in the output stacks on the left side, the negatives on the 2 stacks in the middle, or the 2 output carriers in front of the instrument. And if there is a problem with reading the barcode, for example, or a wrong medium, then the plate will be transported to the error stack. All time you can see the loading of each stack by the loading display in the touch screen, and if a stack is full, it will be indicated by a light signal or also at the touch screen. The most important question for inventing it in this MSR module in the routine diagnostic is how good is this to, and how could it correctly categorize the MRSA screening samples? So we have evaluated the EPIS module twice, once in our laboratory in 2019 when the instrument wasn't there placed for testing, and the second time when we started to implement it in the routine in 2021 at the beginning. For the screening, we have compared the visual reading of the plates by a technician with the EPIS-based categorization. And we are performing our culture based on a Chrome ID MRSA agar, which is designed for reading after an incubation of 20 to 24 hours, and in negative cases, after another incubation time of additional 24 hours. Because most of our samples arrive in our laboratory during the late afternoon or the evening. So when we started reading the plates in the morning, most of them have only an incubation time of 12 to 16 hours, so not reaching the 20 hours. So routinely we are performing a second reading after another 24 hours incubation. As you can see here, we have checked 2,262 samples at one time, at the first time point, and 1,546 samples at both time points. And as you can see here, we have with the second reading get every MRSA. We have also found by the visual inspection, so given a sensitivity of 100%, even the specificity, which is high at the first time point, is declining to 88% in the second reading. But you can see most of the plate, 95% or 87% of the plate, doesn't need a manual handling by an EPIS reading. And this excellent sensitivity we have found is also shown by other study groups, by some ECMID posters or publications. And I show you here 5 independent studies of the last years which have tested more than 16,000 samples for MRSA. And all of these 5 independent studies show a sensitivity, as Steve says, from 100%. So you can pretty sure not to overlook an MRSA. The specificity is better than in our hands in the studies, reaching from 95% to 98%. So we are really convinced not to overlook an MRSA, and so the MSM module is designed for 3 different chromogenic media, and as I show you here, that the sensitivity is not dependent of the use of a special medium. All of the 3 media are working pretty well. And now important, what does it mean in a time-saving point of view? The handling time of true or presumptive positive samples are the same for EPIS-readed or visual inspected plates. You have to have a visual inspection, then you perform confirmation tests or induce an antimicrobial susceptibility test. The really important saving of time happens with the negative plates. Instead of putting one plate from one hand, open up the lid, looking for it, close the plate and put it on the other side, the EPIS categorized plates can directly be discharged without further handling. So for the time the EPIS is running, the technician is free for other work. And that's the main point. Showing 2 exemplary days in our laboratory, only 49 plates instead of 1,046 plates has to be handled by our technicians. And as you can see, she's very happy about it. So altogether, at the moment, we are saving 3 to 4 hours of a technician working time by using the EPIS system for the MSR screening. In the last part, I will show you some steps you have to perform as an EPIS system for maintenance, as well as a workflow, as it works, and some examples of different results. First, in the morning, you have to perform a so-called color check. Because categorizing of colonies are based on special colors expressed, an appropriate color detection must be ensured each day. So using a plate with standardized color dots, you are performing a kind of color standardization each day. Second, you have to perform a so-called system check. Here you are using 2 plates with some different painted colonies. And with these, the recognition and counting of colonies are checked, as well as the transportation of the to the right output stacks. One plate is designed for being sent to the positive and the other for being sent to the negative stack. And both checks need less than 5 minutes daily, and errors in the color check are extremely rare. I can remember only once in our laboratory. The system check fails sometimes, but if you clean the plates and make it again, most often it runs properly. Third check you have to perform is a biological quality control. So an MRSA on the same chromogenic medium inoculated at the same time point when the samples of the patient of the day are inoculated. And this has to be the first reading each morning. So if the color check don't work, if the quality control don't work, so the EPIS is not really, or is not safely identifying an MRSA as an MRSA, at this day, you have to check all the plates manually and then check what the problem, and most often it's not a problem of the EPIS system, it's most often a problem of the chromogenic medium. The company recommends also a negative control. This is an MSSA on the chromogenic medium, but to my opinion, that's not nearly necessary, because if this negative control fails, the MSSA would look like an MRSA. You will find them in the presumptive positive stack and check them anyway. So I think the do not make a negative control. Additionally, you have to clean the instrument on a weekly and monthly basis, and you have to perform a shutdown of the instrument once a week. Cleaning the outer surfaces of the input module, the imaging module, the output module, as well as the touch screen with an alcohol moistened tissue normally needs not more than 15 minutes. Monthly, you have to clean also the inner parts of the instrument, a more detailed imaging station, but this needs not more than 30 minutes. And altogether, so the maintenance of the instrument doesn't need a lot of time. So how is the workflow? When you started a session in the morning, you will come to a so-called session list. And in this session list, you have displayed all the sessions run like you, as you want, all of a day or all of a week or all of a month or the last month. And in the first column is the date of the session. The second column shows you which module in the session was running. So here the infection control is the MSA screening. The session was an MSA screening. and the SC, that's the qual tool, that's the system check, and at this state it was okay, it was run to the positive and also to the negative stack. The infect control research use only is a VRE screening at this moment. If you would like to see the results of a session and to control this, then you click on the desired row, and then you will see the so-called sample list. And the sample list, each sample of a session is explained. And the first here is the 8325 MRSA. That's our biological positive control. And as you can see, it was correctly categorized as positive. And if you want to see it and control it, then you can click on the number, and then the image of the plate will be displayed. And you see here, it's really greenish colonies, green colonies. So the positive control was correct. You can also look for the negative designed plates to see whether they are correctly negative. And so you will find obviously plates with no growth or with not significant or not clearly positive growth. So some colonies, some white colonies, small colonies are clearly negative. If you would like to control the positive, then you can display the positive list of a session. All the positives are here, and you can check each image if you want, and then it will be presented here as the first one, clearly green colonies, and it will here in a report reported as colonies with typical chromogenic intensity for presumptive MRSA. And as you can see in the next examples, these typical green or other colored colonies are expressed not only in small numbers, also in high numbers. So it's clearly a good signal for true positives. The APAS is very, very careful. So even if only one single colony or only slightly changes of the colony at the margin happens, they will find it and they will send it to the positive stack. So it could be very convinced that you will not miss true positives. Sometimes you see here a positive, and you think about and you check it visually, also or by imaging, and say, what does upper think is here positive? And then in this moment, you can let him mark here by touching the colonies and let marks with a square the positive MSA, what he interpret as positive. And you see in the small squares, okay, that are not colonies. You think that are real MRSA. So this is a false positive, but it convinces you that really a true positive you will never miss. Sometimes, okay, and I see also he's very, very careful. So what he marked positive is light greenish color on one colony, or here on some other colonies, or very, very tiny colonies, or colonies on our green labels, which look a little bit greenish. So all you will send as positive, but you can clearly then see very quick that's not a real positive. Also, sometimes you'll find some artifacts as positive, some chihuahuas. You have here growth. In other examples, you have growth. It's a special color, but you see truly that's not a staphylococcal morphology. So also this presumptive positives are when the check you can very rapid see it's negative. And sometimes you have some artifacts which he interpreted as positives. Here our green label is, the corner of the green label here was marked or seen as positive. Sometimes it's a crack in the agar, or sometimes this happens, the knob of the Petri dish is screened or is interpreted as a greenish colony and sent to the positive tags. So these plates you can immediately check as negatives without further time loss. So taking that together, the EPIS independence is a very convincing system for culture-based screening of MRSA samples. And it allows reading of up to 240 plates in one session, and a complete session runs 72 minutes. Reading of the most common used chromogenic media for MRSA are possible, and the plates are sorted based on the special software in presumptive positive or negatives. Based on different studies and in our own hands, the system reaches an excellent sensitivity, and that's the most important, sensitivity of 100% with a high specificity, especially after 24 hours of incubation. And the most important and nice thing is that you really save working time for the technician, especially working time which is boring for the technician, and to save her concentration for the more sophisticated things they have to do. And this is really a nice instrument, and we are happy that we have it. So thank you very much for your attention.
Thank you, Dr. Schumacher, and welcome back, Dr. Giglio. So to our audience, it is now time for our live Q&A session. If you have any questions you'd like to ask, please do so now. Just submit them into the Ask a Question box and click Send. We will answer as many of your questions as we have time for. So it looks like we already have some great questions coming in. Dr. Schumacher, let's start with this one. When you are saying you are saving per day 3 to 4 hours, what does your team do with the free time that they have? Meaning, which other tasks can they focus on, and what effect does it have to your staff?
7 years ago, when I come here to the lab, there were 2 technicians working only for the screening samples. And today is only 1 technician working on the screening place. So, okay, the other technician can do other things. And today, not all screenings are running over the EPIS. It's only the MRSA, and we are trying the VLE screening. And so when the sessions are running in this time, the technician is performing the visual inspection of the screenings for multi-drug resistant gram negatives, which is has to be done visually and by hand only. So this is, we are making half of the technician workload for EPIS.
Very good, thank you. Our next question. What are the 3 key benefits for using the APAS system?
Okay, saving the working time. It's really 100% sensitive. We don't overlook it. And some studies showed that sometimes the EPIS find MRSA which was overlooked by the technician. So this would also be happen. And the main time is the quality of working time. So the technician is not get tired by looking on empty or clearly negative plates 1,000 a day. So she can be concentrated for which she is more trained for doing some more sophisticated works and not looking on negative plates. So this is really quite nice for all.
Certainly. Our next question. Have you included the time for the data transaction while measuring the time saved through APAS?
And I not really understand. Do we measure the time we saved?
I'll repeat.
APAS? Sorry?
Yeah,
I did not.
Have you included the time for the data transaction while measuring the time
Um,
saved through APAS?
the data transmission, I'm not sure what's going on.
Okay,
I'm
maybe
not sure.
we can have our
So,
submitter reword
mhm,
that question
it's-
and resubmit it.
the data transaction report to the list. Okay, unfortunately, and that's not a problem of the APIS system, it's really a problem of our own LIS. We have no connection of the results directly to the LIS. So when this works, at the moment, our technician has to put the negative result per hand on a. So if this will come, and that's really a problem of our LIS, not the Apple system, then we will save, I think, half an hour more time again. So when we have a direct connection to the LIS. And other laboratories have that, and they're quite also happy with that, and with their LIS system it works.
Very good, thank you. And Dr. Giglio, this one's for you. How does the APAS AI use a convolutional neural network architecture?
Very good question, and the answer is no. The type of technology in APAS is very early machine learning, and there's a lot of good application for CNNs in different problems, but for our particular problem in microbiology and bugs growing on plates, the very early machine learning systems do the job. So I think as we look at APAS 2.0 and 3.0, would we look to move to CNNs or other AI, where we get to see a need to do that?
Thank you, Dr. Giglio. Dr. Schumacher, let's go with this one
Mhm.
for you. Do you see an impact on the quality of your result?
Okay, first, our visual inspection has a good quality, and we compared to the visual assessment of the technicians. So the EPAS is in our hands not better, but okay, but you can think about a technician looking on 1,000 plates. She become tired and do not have her concentration. So sometimes, and that doesn't happens with the APIS system. So I think it's a kind of quality control that the APIS is an objective measurement, and you can look at all pictures. So if there are unclear result, or you have a situation you don't know really, then you can look and control with the pictures. And this, for me, it's a quality control. So we can check, and sometimes if a new technician is working, we can check together the plates and make a kind of training with that. So at this point, it's okay. It's more constant quality, and you save the high concentration of the technician for really important plates, the presumptive positive plates, and do not waste it for empty or clearly negative plates.
And Dr. Schumacher, on what is the team focusing more on with no need to read those negative plates anymore?
What they are doing instead, okay, reading other plates, not empty plates. So for the gram-negative screenings, which are not so easy at MRSA. It's one column upon one kind of color on an agar. So the gram-negatives are quite diverse. So this is more at the moment, it's only can be done by a visual inspection. And this is the main time our technician on the screening place is doing.
Thank you. And then it looks like we have time for one more question. We'll wrap with this one. Do you see future potential with APAS with other workflows?
Oh, yeah. um, okay, we are looking for the VRE screening because the second one, okay, if it works for the gram negatives, I would be very happy. But what's the more interesting thing is we are working on the reading of an agar diffusion test, because yes, imaging and the imaging of the diameters of the disk diffusion plates, and then you can interpret it. And this is also a more objective reading than by a technician, which is often only they assumed, okay, that would be sensitive or not. So this could be a very fine system to use it for reading agar diffusion tests, especially if you're not possible to buy a quite expensive total lab automatization, which can do that also. But this is with much less effort. You have good, and I see one study where the MRSA reading in APAS was better than in the total lab automatization. So you can have that in a quite more not so expensive form with a high quality. So, and then I can think about reading the agar diffusion test and then making the screening test. So the system will be can be used for multiple functions. So that's my thing I would like to see in the near future.
Thank you, Dr. Schumacher. And thank you, Dr. Giglio, for both of you for your time today and your important research and your excellent presentations. Do either of you have any final comments for our audience?
Try it. I really can. I would support you. It's a really good machine for that. It will help you.
Excellent.
I do not get any money from that. It's really I have to say, it's my own opinion.
And Dr. Giglio, anything you would like to add?
I think it's been great listening to Ulrike, and I think Ulrike, you may or may not know, but we are doing the disk diffusion testing, and we have published some work with Dr. Wisplinghoff and also Lisa Brenton from St. Vincent's in Melbourne, which is a development module, but we are looking to solve that problem, and so far it's working quite well.
Very good. Thank you both again. And we'd like to thank Labroots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Now, before we go, I do want to thank our audience for joining us today and for their interesting questions we were unable to answer today due to time, and also those that will be submitted during the on-demand period. They will be answered via the email address you provided at your time of registration. Today's webcast can be viewed on demand, and Labroots will alert you via email when it's available for replay. We encourage you to share that with your colleagues who may have missed today's live event. Thank you again for joining us. That's all for now. Have a great day.
Thank you.